Thank you very much, Dawn, and the Dale Travis family had a delightful dinner tonight. And when you were making that introduction, I was wondering who's making this speech tonight, because I wasn't sure I recognized all those wonderful things. But thank you very much. It's a pleasure to have such a wonderful group of people, all of whom are interested in our lake. This talk is titled, or subtitled, Effective Collaboration Among Students and Scientists, Agencies, and Private Enterprise. And really, it is about us. It's about all the people in the room, many people throughout the state of New York that have contributed to our knowledge and our love, really, of Onondaga Lake. It's a small lake, really, of 2,900 acres, only about 12 meters deep, maximum depth of about 20. Turnover, the amount of water that comes into the lake per year, it turns over as much as 5 times a year. Actually saved the lake in many times because it's a relatively short residence time. The basin was probably carved about 15,000 years ago, filled with glacial water about 10,000 years ago. As we know, and from the image, we remind ourselves of how it is impinged upon by the surrounding city of Syracuse. Tonight, we will describe a little bit of the history of the watershed, a few of the legal highlights that one needs to be aware of as we begin to study and understand the lake, the water quality responses to its being perturbed over the last couple of hundred years, and then renewal, at least the beginning of renewal, our knowledge, the future organisms living in the lake, and collaboration secrets that we may have begun to see emerging as we work together. Environmental Science and Forestry College is on the land of the Haudenosaunee Confederacy, more than 1,000 years old. It's extremely important land. The Heiberg Forest, also the Tully Forest, is also their original homeland. And the term Iroquois was used, or has been used, to describe the 6 nations on Onondaga Lake's shoreline. Europeans arrived in 1654, in particular Father Simon Le Moyne, and he recognized the importance of the lake to the Haudenosaunee Confederacy and the proximity of the salt springs in this region, a remarkable and rather unique region in the United States. And historical records such as by John Bertram in his travels recognize the lake rich in salmon trout, which were probably Atlantic salmon, and rich in eels that could be readily captured. This was way back in 1751. Ben Franklin recognized what he called, or he thought of as, the Romans of the New World, the Iroquois, extremely important as colonial statesmen. And in fact, in 1776, the chiefs of the Iroquois nation were invited to the Continental Congress. The conceptions that we have of a democracy emerged in this region. Sainte Marie among the Iroquois, many of us has visited, but it has now become Skä-noñh, the Great Law of Peace Center, and it has just opened in November of this year. And so the emphasis now is not so much on the Europeans among the Iroquois, but rather the Native Americans themselves and the phenomenal activities and successes that they had, and we believe are continuing to have right now. Syracuse was known, still known, sometimes as Salt City. Those of us with cars that are all rusted out know that very well. But way back in the Revolutionary War period, commercial salt production in the late 1700s was extremely important. Production of salt as many as 9 million bushels in the late 1800s. The major North American supply of salt came from our region during the Civil War. In 1822, what we would call today wetlands, but they were called then swamps, were drained, and the lake was actually lowered, changed its shape as a result of that. The Oswego Canal was built in 1828. Late 1800s to 1900s, the lake was a marvelous place, at least if you're interested in recreation and fulfillment in terms of physical outdoor kinds of things. And the sketch here almost looks like one of those menu pads that you see at some of the smaller restaurants where you look down and there's all these activities, fishing and hunting and boating and sailing. And that's what was going on in Onondaga Lake. 8 major resorts, late 1800s, early 1900s. And here's just one of them, 1906 to 1915. Shoot the Chutes. Wouldn't it be great if we had one of those today? But this is a spectacular example of what was there more than 100 years ago. Industry began to evolve, particularly with the soda ash factory, moved here from Europe in 1886. Ice harvesting was very important in the lake. That's what we put in our ice boxes up until about 1901. Sewer system was developed, salt production ceased, and by 1940 we were not swimming in Onondaga Lake anymore. So you could say that the system was perturbed, which means deviation of a system or a process from its normal state or path, particularly caused by an outside influence. So I think it's a pretty good term, a satisfactory term for Onondaga Lake ecosystem. Seems like wherever I go, I work with perturbed ecosystems. I grew up in California, then went to Oregon, Michigan for studies, British Columbia, and of course now in New York, but also in Russia and Spain and Mexico. And all of those places have been perturbed, but perhaps not to the extent that our Onondaga Lake was. From 1920 to 1950, this region was a phenomenal manufacturing center. Not only the salt industry, but also Onondaga Pottery Company. There are still large pieces of pottery along the northeast shore of Onondaga Lake. There were typewriter shops where we've actually had to spend millions of dollars cleaning up after the typewriter materials, for example, when the Center of Excellence was built recently. And there were many car manufacturers such as Franklin. Franklin engines still fly over Syracuse in airplanes of today. Very important event was the development of New York's canals, proposed in 1699, completed in 1825. So you sort of think, I don't know, we can't get anything done. It takes forever. And here was an extremely important, successful deal. But I learned this morning at a meeting for the biennial canal conference that's coming up a year from now that it was 1817 when the first shovel-ready, actual shovels were dug for the Erie Canal, and that was completed in the first piece by 1819. But we mentioned before that in 1822 the water level of the lake was lowered. And eventually, though the canal system remains and is very important recreationally in New York, the railroads began to outcompete the canal system, and of course later on trucking tended to do the same to the trains. But it's phenomenal that on the menus in New York City was Onondaga Lake whitefish. Probably was a cisco, a different species than our whitefish, but nonetheless it was a famous dish at that time. And we'll talk a little bit more about some of these other fish a little bit later. The water quality responded to these perturbations dramatically. And in a way, the Onondaga Lake system mirrors worldwide environmental changes. In those various places that I mentioned, almost any continent in the world today has these kinds of fishery changes in their aquatic systems. One of the biggest effects was the soda ash factory, literally opened for a century, the largest in the world, probably helped us solve or help win World War II because of soaps, ammunition, and other things developed needing soda ash. But the output was Onco lights, which we called at a conference I was at in 1990, false rocks. They're very light in density, they're made of calcium carbonate, and they dominate the northeastern shore of the lake. There are still today combined sewer overflows hundreds of miles of sewer line that mixes stormwater and sewage water and still discharges high levels of nutrients and chlorinated hydrocarbons, although things are better today than they were. But if you were to make a checklist of Onondaga Lake at its peak, many of us probably saw the movie Civil Action, and especially Aaron Brockovich. It wasn't so different from that. Dissolved oxygen levels plummeted. Nitrogen, phosphorus, and potassium were at levels that were basically almost like fertilizer. Calcium carbonate, sodium chloride, low visibility, mercury, zinc, substrate. Whoa, this place really got a dose of many of these compounds through its history. And dissolved oxygen especially dramatic in the lower end of the lake in the summertime, at least 5 months. There was no oxygen there. And even in the upper portions of the lake in the fall, when mixing occurred, there was no oxygen, except in a very few refuge areas. We found back in the 90s that radio tagged fish left the lake in the autumn, and many of them did actually return when the oxygen in the lake was suitable. Legal highlights, we'll just mention a few of them, but we need to have a little bit of backdrop as far as the ways that we've begun to try to deal with these effects. The Attorney General sued Allied at the time over the mercury pollution. Fishing was banned in 1972. New York State DEC joined the suit in 1989. A very important Onondaga Lake Management Conference was developed by Moynihan and Walsh. But by 1993, at least there was an actual plan of action for helping the lake recover. By 1994, there were actually 12 subsites described in a Superfund system, which would eventually provide some additional funding to understand and save the lake. 97, 98 was an amended consent judgment. 1999, the ban on eating of fish was lifted, at least for some sizes and species. And by 2007, federal court approved a plan for Honeywell International to remediate the sediments. So in 1986, the soda ash facility closed. And in some ways, that was the beginning of renewal, although I really believe that much of the legal work that was going on was essential to making some of this happen. And when that happened, the salinity level of the lake, sodium chloride and also calcium carbonate, dropped from about a level of roughly 10% of seawater to a more normal level, or basically was cut down to a third, right when that occurred. And in 1986, serendipity. Chris Gandino, sitting over here, decided he needed to do a term paper. He was getting his bachelor's degree in environmental studies. He wanted a term paper. I didn't want him to take a boat out on the lake. It might hurt the boat. So a gill net in a wheelbarrow was what we did, or what he did. And that term paper assignment, I realized, oh my gosh, there's fish out there. And there's not just one species or two species. There were a number of species of fish. And Chris went on to do a larger project. He went on and eventually he did his master's degree with us. He's now in charge of the ambient monitoring program for Onondaga Lake. He just loves the place. But in any case, it was serendipity. I didn't even know at that time that the soda ash facility had closed. It was just that year. And a little context, the gasoline was only 89 cents a gallon. Ronald Reagan was president. And sadly, the space shuttle disaster occurred in that year. But Chris was the beginning, really, of an academic family that's gone on for literally 30 years working on the system. You noticed on the slide on the right, happy people. If you feed people, they're happy. Dale Travis figured that out for us and Dr. Leopold today. But they are a wonderful family. On the left there in the boat is Patty Thompson. Patty, I saw you a minute ago smiling. There you are. So the students, this talk is really about the students and the work that they have accomplished on the lake. And here are most of them. I fear that I've missed one or two. Most of them, I've had about 75 graduate students, and usually they've done something in the lake, but these folks actually got their degrees associated with the lake. Some primarily on fish, some primarily on invertebrates like crayfish or dragonflies, but often both. And very importantly, 2 to 4 undergraduates per year have contributed to the lake since 1986. Now, we were a little bit on the ivory tower mode at the time. We didn't have any special money. We had peanut butter and jelly. We still work with peanut butter and jelly, but in any case, we were just out there studying the lake because we were interested. And we had preparation that ranged from something sensible like fishery biology, general zoology, aquatic entomology, but we all studied together. We were just into learning about nature and about animals and a little bit about aquatic plants and aquatic invertebrates. And in some ways, perhaps some of you have set up aquaria at home and you get this little booklet and it says, okay, you got to have a balanced aquarium. I mean, what is a balanced? Well, a certain amount of algae, a certain number of fish feeding on the animals that are living there, some bigger fish, and so on. And in a way, that's what we are trying to do with Onondaga Lake, is to understand it well enough to set it up, to help it return to a normal functioning ecosystem. Here is an undergraduate who's now worked then, is now a veterinarian, worked actually with Patty Thompson. I don't know, maybe she didn't like working with all of that algae, but this is how rich the lake was in the late 80s, early 90s. And we had a conference at Lake George. A bunch of fishery biologists and limnologists got together at Lake George. We didn't even know which fork you were supposed to use, or spoon, you know, it was a fancy place at Lake George. We had a great conference. And we wrote a simple 8-page restoration document about that we felt the hypolimnion, we could see it was off limits. Nobody was going to live there, at least in the fish area. Fish nesting was limited. Waves and oncolites inhibited the macrophytes that would normally be found, aquatic plants. Juvenile fish success was extremely limited. And we felt that wetlands would contribute to fish habitat if there were any nearby. So I said, oh, well, I'll just order some gravel and we'll build some artificial spawning sites. I mean, a fishery biologist, aquatic biologist should know how to buy gravel, right? So the truck arrives, oh my God. So in any case, we shoveled this. We didn't have anything fancy other than shovels and a little boat, and created some spawning sites and eventually some wind-protected habitats with hay bales, as a matter of fact. So it was pretty basic. Here's Chris Gandino. He wasn't necessarily always, no, he was always excellent, but we just shoveled this stuff in. Mark Arrigo. Mark, are you here tonight? Mark Arrigo is with Parsons, and he was very prominent, is prominent with our studies even today. But basically, we set up these sites to see if the animals would spawn there. And they sure did. And they were attracted even to a stake pounded into the gravel, because there was almost no cover. There was no substrate, no cover associated with the lake at that time. And we experimented. Don, I don't know if you realize that we planted, I think, as many as 12 species of aquatic plants, because we didn't know that the seed bank in the lake could actually seed these plants. We didn't realize that. So we bought all these from a company in Wisconsin, and we had contests who could plant the most. And Dr. Madsen, very significant scientist, had carpal tunnel for years as a result of that contest. So here are some very happy campers as well. Steve Burgess is Dr. Robert Burgess's son, worked with us all summer. Dr. Burgess was a chair of our department. But the activities that we're doing today don't look terribly different from those that were done in those late 90s. And from the air, we have in the upper left there a picture of hay bales, which help to protect the wind action on these sites. And then some enclosures to protect our planted plants from carp, because we didn't want them all torn up. There are folks in this sketch who I won't go over them all, but some of them, almost all of them, are aquatic biologists now. There are some that look like they're 12 years old. It's fantastic as you start to look at the data. But we'll talk more about the fish in a few minutes here. A closer view installed in 1993. The interesting things about these enclosures and structures is that Honeywell used much of our findings when they set up the plan to not only dredge the lake, but put a cap in that had habitat. So we were very excited later on when the record of decision was made and they went forward with habitat structures that really spun off much of the work that we had done back in a decade before. Now, we did have running water, but we didn't have PowerPoint. I don't know how we lived. I really can't picture life without PowerPoint. But here's an example of a slide that we would have projected. It probably took an entire afternoon, if not a whole day, to make such a slide. But it's an important piece of information. It shows that when we manipulated these sites with regions that block the waves, when we put spawning gravel in, tremendous effect on sunfish nest density. So we saw the lake had potential. It just needed some additional help. So a lot happened between the 90s and 2000, and in particular a marvelous book that I shouldn't say it was edited by Steve Effler. I should say it was cajoled by Steve Effler. Marvelous scientist, Upstate Freshwater Institute, in charge of it for many, many years. And that book has been a seminal accomplishment. But a lot has happened since that in terms of phosphorus and ammonia changes, zebra mussels, zooplankton shifts, and recruitment of fishes. So we'll cover some of those things before they drag me off the stage tonight. And one of the most significant things that happened was an explosion in the population density of zebra mussels. Zebra mussels, if you look at the figure on the lower right there, you see 221, 50, 32. There were high densities of young larval zebra mussels in the river, the Seneca River, but they fade out as you go further and further toward the southern part of the lake. And basically, we're pretty sure now that the high levels of ammonia that were in the lake at that time kept them from being successful. And the year that the county managed to deal with the ammonia, they exploded. And we had tremendous densities. And in fact, Mike Spada got his master's and then this publication as a result of that recognition. So between about 1999 and 2000 was a tremendous reduction in ammonia. That's when zebra mussels took off, and now the ammonia levels are really quite low. One of the things we worked on very hard was some structure to put into the lake. And we actually had a really great plan. We wanted 4 sites in the lake that would have basically breakwaters and protected areas and gravel that fish would spawn in. 4 sites, but we only got one. The arrow that's moving there is in the northwest portion of the lake. How many have ever seen what is called the permanent habitat module? It looks like a breakwater out there. How many have actually seen it? Not too bad. I'd say, Patty, 10% of us or so have seen that. When that went in, Patty called me up and said, they're putting it in. Oh my God, we didn't know. In any case, it was built. We assessed it as best we could. And we weren't hugely impressed with the outcome. The lower left there shows an artificial break that we created, and then this was built. But it was probably built a little too close to shore. It was built exactly opposite a reconnected wetland. And so it was a tough thing to assess. Probably did not greatly enhance the community, we thought. But now we realize that further away from that structure may be some very important habitat, particularly aquatic plants, and this fish came from there. So we're pretty sure that we might have been too close physically to the structure to fully assess it. Phytoplankton lake-wide have diminished tremendously in response to changes in ammonia and particularly in phosphorus. We know a little bit about the composition of those plankton. We don't have the blue-green algae. I remember my mother-in-law driving past it and says, oh, it smells like BO. It did. It was awful, but it's much nicer today. The phosphorus has plummeted between 1990 and 2012. Alewife are an introduced species as a result of the canal system. They modify the size of zooplankton. They forage so effectively. And when there are many of them in years where they're very abundant, the size distribution of the zooplankton plummets, and that has effects in turn on the phytoplankton. Benthic macroinvertebrates are the creatures that are not fish, that live on the bottom like dragonflies and crayfish and so on. And some people would just say, well, that's fish food. And in part, that's true. But one of the significant things that we have found is that has been little change in 25 years. 25 years ago, Bruce Wagner and I collected samples. The samples that we get today are not greatly different from those. So in a sense, we could call benthic macroinvertebrates an example of hope, but not very much change. Around 7 families has just stayed for those 25 years. Brandy Brown, working with Kim Schultz and I, did some field experiments and some lab experiments and found that in some cases the substrate, the animals, the mayflies, could not live on the substrate that we took from the lake, even though other lakes we got good survivorship. And she did work in the laboratory and found that the Onondaga Lake substrate, in some cases, the survivorship was quite a bit less, only 52%, in contrast to other waters where it was much, much higher. So we're pretty sure that the benthic community is having some issues even today. More recently, Zach Smith defended his master's degree, and he decided that it would be valuable to know, well, okay, if things are a little sparse in the benthic animals in Onondaga Lake, how does that compare to Otisco and Cazenovia lakes? Sure enough, believe it or not, we'd never done this before. No one had, to my knowledge. But yes, the benthic diversity, number of genera was less in the Onondaga Lake versus these other lakes, conspicuously so. One could look at this as a biological chance to respond, in that we are remediating, in particular, the southern portion of the lake. And one of the things that Zach did with his thesis is that he looked at the number of genera, the kinds of animals outside the remediation areas, more in the northerly areas, versus inside. And he found that, sure enough, the areas that were about to be remediated, and now they have been, had fewer animals in them. So the optimist in me says, okay, it looks like we have a really good shot then at seeing those numerics increase, at seeing the diversity increase as a result of that remediation process. We studied snails much more recently. This last year, there were 18 species of snails in Onondaga Lake based on the shells that we collected. Nobody was home, but based on the shells. 11 of those are not in the lake anymore, 11 species. 4 of the species that were lost actually are still found in the outlet of the lake. And we've actually moved some of those into the lake, put them in enclosures, and we find that actually they can survive. So they might be returning at a snail's pace, not sure, couldn't resist saying that. But in any case, there are some interesting features about the benthic animals in this lake. The aquatic plants, or macrophytes, and the epiphytic invertebrates that live on them flourish in Onondaga Lake today. That's a fact. Decreased nutrient loading, coupled with the introduction of zebra mussels that has cleared the column and allowed sunlight to reach these aquatic plants and allow them to thrive, has enhanced the aquatic vegetation dramatically. We've moved from 5 species when we first started our studies to 23 species in only a dozen years. From 12% coverage of the littoral zone to 80% coverage by aquatic plants. And this is one of those slides, don't read it, don't look at it, but you can see a dramatic change here that Lucas Kirby just did this work, where it's clear that a new species colonized just last year, Nitylopsis obtusa, which is an exotic species, invasive species, is now #1 in just 2 years. So the aquatic plant communities are extremely dynamic in Onondaga Lake. They're not only growing, they are changing. Living on the surface of plants, the term is epiphytic. The invertebrates there are more diverse than the benthic. There are many more species of epiphytic invertebrates than benthic ones. It's still lower than other, the epiphytic invertebrates in other lakes are still higher than those in Onondaga, but it's still an encouraging sign. Here is a busy slide, but the epiphytics you can see are basically more families are found in Onondaga Lake, but waters adjacent to Onondaga, such as Otisco and the Seneca River, far more. So there's still a depression in these invertebrates so far. Well, now we get to talk about fish. And I told Don I wouldn't go over, but I fibbed, because it's going to take at least 2 hours to talk about fish. How many of you are fisher persons? Wow, I'd say about 80% or so. This is great. So if they're not biting, you could use an electroshocker. But we use trap nets as well, seine nets, gill nets, and we use a consistent approach around the lake, and we have for actually quite a long time. And it's important that there have always been fish in Onondaga Lake in modern times. There's never been fewer than 12 species since 1927. They don't look like this. They look good. They're not bug-eyed. They're not tumored. They're generally in quite good, remarkably beautiful specimens. 64 kinds of fish we have seen since we started. So we've got now a 30-year run that we don't get 64 every summer. We might get 40. But we've seen a total of 64 over the years. We've gained some species. I mentioned alewives, not necessarily a positive effect. But carp were introduced deliberately, and there are gizzard shad, sheep's head, and others that have reached it almost certainly by way of the canal system, except for carp. About 20 kinds of fish spawn in Onondaga Lake. So waters that are good enough for natural reproduction to occur generally have hope. If nobody spawned in there and they only came from the Seneca River, that would be a tough situation to be working with. We have 3,000 of these pie charts, but all this shows you is that you don't get the exact same distribution of fish from summer to summer, but by and large, the kinds of fish that dominate include not only the sunfishes, but also in some years, planktivorous fishes, such as gizzard shad and alewife. What's very important to aquatic scientists, fishery biologists, is the number of young fish that come into a population. We call it recruitment, if we're paying tuition for the term. And part of Patty Thompson's work was to look at the young bass numbers, and you can just see from the pattern there, from 1994 to 2004, there's a lot of variability. But generally, the part of the lake that's good stays good. The part of the lake that isn't good, that it has no dots, no young bass spawning, stays that way. I think that we're going to find that that southern part of the lake, as it's remediated, in fact, will very likely spring forward with bass reproduction. But the sunfish nests vary annually, not just because it's Onondaga Lake, but this is what happens in the Northeast at least. 10,000 nests in 2007. 2,000 in 2009. We just started doing this after my sabbatical. I went to British Columbia to study sticklebacks, and I said maybe we ought to be counting the nests in Onondaga and use that as part of our recruitment. And we've been doing it ever since. And the county, through the ambient monitoring program, has as well. A few fish have received special attention. They're all important, love them all, but we've focused on some. Bass. This is Lucas Kirby completely dominating a contest using the shocker. We didn't mention the people at the contest that we... But the growth rates, condition, and size distribution of the fish in the North Basin and the South Basin are not very different. They're really very similar. The densities are different. There are usually more fish in the Northern Basin. Lake sturgeon are the largest fish in the lake, up to 1 inch shorter than 6 feet. Have we beaten that yet, Lucas? We're still there. Okay, they're getting smaller, but in any case, they're taller than than a lot of us are. And I have to admit that I have a few more pictures of students holding fish with smiles, that is, the students with a smile, than are necessary to drive home the point, because one of the real points of our studies is that many people have learned about ecology and ecological sciences as a result of this work. So Rachel and Eric are holding these fish, and we've handled quite a few. Danielle Hurley. Daniel, where are you? I saw you way back there. Anyway, she's the world's expert on bass, but also she's working on the sturgeon. And you'll see that in 2013, we actually captured 35 of these great big sturgeon in Onondaga Lake, working also with the U.S. Fish and Wildlife, tagged fish, and so on. Extremely exciting. 24-hour sonic survey. Here's just an example of a walleye. One-eyed jack is not the name that we appended to Stephanie Johnson. That's actually, the fish was called one-eyed jack. But notice her outfit. I think we didn't want to be recognized by. But it's fascinating to look at the distribution and the movement patterns of these walleye. It's just incredible. 8,000 meters travel. Here's another set of data that just shows that by and large they tend to be in the upper 2/3 of the lake. A couple different years we have. But the fish in the sketch there was caught in Sylvan Beach. It was tagged by us, and it was just reported. Eric got the call March 15th, and the fellow ate the fish. And I guess it was very good. There are brown trout in Onondaga Lake. How many have ever caught a brown trout this big? Awesome. How many would like to? A beautiful, beautiful fish. I have the idea that eventually someone's going to learn how to catch these in Onondaga Lake, and it'll be a very exciting thing to do. But what Curtis Karbowski did in concert with Mark Thies and also Karin Limburg was to use some sophisticated tools to figure out the movements of these fish into the streams, what their chemistry of their otoliths and their scales was. This sketch just shows that there's a time of the year, even now, where there's not enough oxygen and too high a temperature for the brown trout to survive in the lake. They survive, they just don't stay in the lake. They go up some of the streams. And so the techniques that were used there, again in concert with Karin and Mark Thies, were to look at stable isotopes in the scales of the otoliths and basically follow these animals. Here are you'll remember the otolith maps that Karin showed in the Dale Travis lecture a while back. These Curtis did for his, but it basically helped us understand what streams the fish might be going to or might not be going to. Now, Steph Johnson, are you in the room, Steph? Hi, I didn't see you. She says absolutely accurately that you can't just study the lake. The tributaries are extremely important, and many of us grew up as stream biologists, and she's absolutely right. She's now a big player, along with Ed Miklenko, in the Onondaga Environmental Institute. But this is the only sketch I can show you of her work right now. But she basically linked the gradients in the streams with the invertebrates and fish in those streams and helped us understand at least a beginning template of how we need to study those tributaries to the lake. Now, most of us are quite aware of the enormous amount of work that's going on in the lake that Honeywell is doing along with Parsons and QEA. So a $500 million program, especially targeted at Mercury. We see this out on the lake. Most of this activity now is completed. There are some repairs on the cap and so on, but basically the lake is, by and large, that work is coming to a conclusion. One of the features in the lake is that oxygen is very sparse, even today, in the deep areas of the lake. And I was going to be involved in an experiment where we put oxygen in the lake and then look at the response of the fish. And then Upstate Freshwater Institute realized, it's a really fascinating sleuthing, that if you added nitrate to the lake, it would do the same thing as oxygen would do, keeping the mercury from being methylated. Methylated mercury is highly toxic. And also it was more controllable, and it would not stir up the bottom the way oxygenation would do. And it's turned out that the pilot test demonstrated that nitrate addition could be implemented and sufficiently flexible. And we're using that right now on the lake very successfully. And we think that it will help reduce the concentrations of mercury in fish. How are things going to look in our relatively immediate future, but also much longer than that? Fishery, a population, a community of fishes along with its anglers. Here's Patty Thompson and Chris Akios interviewing anglers about the lake. And there have been some marvelous events out on the lake to get people familiar with it as an ecosystem and as a fishery. There are people who go out there in their spare time and are very successful. My son Justin was also on a crew with Chris Gandino back in the day, but he's a very avid bass fisherman. I purposely set up this picture so that my fish would be closer to the camera and might look a little bit bigger. But that was January 10th, an El Nino year that we have. So there is a good fishery right now for bass in our system. Recently I went to a meeting in which I met some people from Burma who fish the lake all the time, and they take the fish home and eat them. Here's a lad on the right, come from North America, has spent many thousands of dollars to go fishing in nearby Thailand, fishing for mazer, completely different operation from the natives on the left that are fishing in Myanmar, because that's what they do. And when they come to the United States as refugees, that's what they do. They catch fish and they eat them. Something to think about. We are very interested in walleye right now, in studying them much more carefully the way we did our bass and our sturgeon. And we would like to be able to eat them, because that's what you do with walleye. They're not a kind of a catch and release kind of a creature. Mercury levels are going down in the fish, but they're still too high, at least in some species, and particularly for large individuals. Here's an example. It's 2011, but it doesn't look very different right now for large fish. Once you get over 400 millimeters, clearly you're above the consumption advisory. That's the fish, by the way, that Lucas is holding that wound up at Sylvan Beach several years later. The health advisory. Has anyone ever looked at the health advisory for Onondaga Lake fishes or for our regional fishes? How many have actually looked it up? Wow, that's excellent. Okay, so I interpreted it as follows, that we're not going to be eating, should not eat walleye, carp, channel catfish, white perch. We can eat bullhead and pumpkin seeds 4 times a month. Even largemouth bass, you should not eat them unless they're smaller than 15 inches. The typical fisher person for bass, not the people from Burma, but people from our region, would probably not keep a fish of that size. Women are not, they should not be eating fish if they're less than 50 years old, and young people younger than 15 years old should not eat fish from Onondaga Lake. No fish. I'm almost to the end now, but I wanted to mention Lake Ontario and its tributaries, because Onondaga Lake really is a tributary. It's on the Oswego River system. And Atlantic salmon were one of the most spectacular species in our region before we polluted Onondaga Lake. And that wasn't necessarily, the loss of those fish was going on all over because of logging, sedimentation, and so on. So it wasn't necessarily all about soda ash or mercury. The last catch of natives was in 1898. We started stocking, trying to recover Atlantic salmon in the Lake Ontario ecosystem, 1873 onward. 2 of my students, just finishing up, Justin defended his thesis on Monday. Chris Powers will defend on next month. I have 5 other students that have studied Atlantic salmon, so it's one of the animals that we really are interested in. It's a native salmonid here. We have taken a few of those fish. I should go back and indicate that that study is supported by New York Sea Grant. And we had a few fish left over that we put in 9 Mile Creek, a direct tributary to Onondaga Lake, in 2014-15, and we're holding them right now in Carpenter's Brook Hatchery, and we'll stock them in about a month. And they will grow and they will live. We have not reached the point where we're seeing them thrive in Onondaga Lake just yet, but it is the beginning for the bringing back of a native fish. It may take a long time. It may always require hatchery production. We don't know that, but we're giving it a shot. We do know that brown trout live there quite successfully. And in some ways, the Atlantic salmon, because their thermal regime is not very different from brown trout, it may bode well for them. Hard structure evaluation is on our compass for this summer. We're going to be putting in a number of structures. Honeywell is required to do this. We're working with them to analyze what happens when we put in these structures, rocks, wood, in terms of attracting anglers to the site, fish utilization, and fish food colonization. So that's a big item that we're working on, a couple of thesis projects this summer. Now, my provost, Dr. Luzatis, reminds me that things aren't just all about fish. And it's so true. In fact, my first boss in Wyoming said, Ringler, worry about the people, the fish will take care of themselves. So, I mean, clearly the people aspect is enormous here. So that brings us to secrets of collaboration. And not secrets that I discovered myself, but watched happen together. But teams are jewels, in a way, very much, just as our lake is becoming a jewel. Strong academic environment at ESF has allowed us to do kind of these things, along with a very earnest community locally. We've had consulting opportunities that we just turned around and said, let's support students rather than be paid as consultants. Our students have become professionals, and they work very well with professionals as they grow through their system. And 30 years of work in the area helped a little bit too. Serendipity counts, again. This, for me, started as a term paper with Chris Gandino. Evolution takes time. Native Americans met here in about 1007, but they were here way before that. 1986 was a special year, 2016, here we are right now. But certainly everything in this lake is changing very fast. And one of the things that's changing the most for me, and maybe for all of us, is the perception of that system, a way of regarding or interpreting something, a mental impression of our lake and its condition. Brings us to the celebrity swim-in Onondaga Lake last July. Out in the lake, nobody was walking on the sediments, but it was regarded in many ways as a success. EPA said that we perhaps should be careful they're not sending the wrong message, that it is true the lake isn't all done yet. But we've had some great success, and I think that that's important. And we have recently received funding to develop programs for the Onondaga Lake system at a science center that will be on the inner harbor. We're very excited about that as that develops and we can solve some of the problems, not only the lake itself, but the interaction and perceptions and working together. And finally, the teams that we've created or that have existed here are based on the students themselves, the professionals, and all of these different groups that I think I've mentioned throughout, except maybe the people in the state fair. But working together, they're the ones that really deserve the acknowledgments and the credit for what's happened on the lake. Thank you.
Time for about 5 minutes of questions. If you want to raise your hand, we'll call on you. Speak as loudly as possible.
How long would it take a sturgeon to be almost 6 feet long?
20 years? Yeah, oh sure, sorry. Yeah. Lucas and Steph, would you say 20 years or more? 15 or 20? We'll know that answer fairly soon. Yeah, thank you. Great question. This is like my class this morning. I couldn't get it going. Oh, okay.
Can you comment, what are the current trends in Mercury in the lake, and can you speak to some of the other contaminants? As I know the fish advisories are based on not only on mercury but also dioxin and I think PCBs.
Yeah, I honestly can't blast all those data out for you. There is a reduction in each of these, but I can't say that they're equal, and I really don't have that data set in my head. Like to.
What do you think is the key to improving the amount of benthic formula?
Well, that's important. We think that we're on the right track in terms of now watching what happens in the southern end of the lake. And we've had experiments where we're actually ready to introduce them, the way Kim Schultz did and Brandy Brown did, actually introduce, because in some ways, like the snails, we think that in some cases they would be there if they could just physically get there. We're waiting around for a bird to drop them or whatever. So I think one way that we can do it is to actually introduce, the way we reintroduce fish to a lake that's been drawn down. But that is a key thing that we're trying to understand. Oh, well.
What is happening to the
You know, I thought about that this morning when I was, the oncolites are still there. They haven't gone away. What's interesting is we used to think they were more of a hindrance, I think, than they really are, because what I think really was going on was the water was so cloudy that the aquatic plants couldn't succeed there. But they're still there. If you go along the White walking trail near Willow Bay, they're there. Yeah, they're not toxic or anything. Hi.
I'm from the 9 Mile Creek Conservation Council, so we're obviously interested in 9 Mile Creek. I'm just interested in oxygen levels in 9 Mile, let's say from the aqueduct down. Is there an opportunity in the future to create fish habitat or pools, woody debris that might enhance trout production? Or
Well, often trout production can be increased with habitat. I'd be very surprised if the 9 Mile Creek is oxygen poor. I haven't measured it there, but usually streams are turbulent enough with it, unless it's really polluted, which it's not. So I think it might be more about structure. Also, there's a fair sedimentation problem down in that part of it. But further up near Marcellus Falls, one of the most productive patches of trout stream anywhere in New York State.
What are the prospects for regulating hydromuscular?
Oh, thank you very much. Obviously, you haven't read your paper. I got my biggest fish I'd ever caught December 1, giant northern pike. I mean, for me, it felt like a muscalon. So they're doing better. John Farrell is working on, I think, in concert with Dr. Leopold, on some spawning locations for northern pike. There are, now compared to 10 years ago, there are far more northern pike in the system. We haven't focused on them, but I sort of fell in love with them after December 1. So
How many more years will you need to inject nitrate in order to prevent mercury?
I think that I saw Dave Matthews back there. Dave, are you back there? Thank you. Dave is the world's expert on this.
Into the foreseeable future. It's not going to end anytime soon, but we're going to evaluate the need for it on a year-by-year basis.
One more question.
So what is a long-term effect of that nitrate addition on the cold water fishery returning to Onondaga Lake?
There is none.
I love this guy. We can talk afterwards.
Okay, so I have one more thing I need to do. We're for every speaker, because we don't give them any money, we give them a very nice bowl, and it's made by a local craftsman, Thomas Horgan, down in the Preble area. This bowl's especially nice because he textured the outside rim and it looks like fish scales.
I can understand that.
But anyway, I want to thank Neil for his presentation.